Dear viewers, this video is part 6 in the 6-part series on the cell and its functions in the Medicine for Common Man series. Types of vesicles formed by the Golgi apparatus, the secretory vesicles and lysosomes. In a highly secretory cell, the vesicles formed by the Golgi apparatus are mainly secretory vesicles containing proteins that are secreted through the surface of the cell membrane. These secretory vesicles first diffuse to the cell membrane and then fuse with it and empty their substances to the exterior by the mechanism called exocytosis. Exocytosis in most cases is stimulated by entry of calcium ions into the cell. Calcium ions interact with the vesicular membrane and causes its fusion with the cell membrane, followed by exocytosis, that is opening of the membrane's outer surface and extrusion of its contents outside the cell. Some vesicles, however, are destined for intracellular use. Use of intracellular vesicles to replenish cellular membranes. Some intracellular vesicles formed by the Golgi apparatus fuse with the cell membrane or with the membranes of intracellular structures such as mitochondria and even the endoplasmic reticulum. This fusion increases the expanse of these membranes and replenishes the membranes as they are used up. For example, the cell membrane loses much of its substance every time it forms a phagocytic or pinocytotic vesicle, and the vesicular membranes of the Golgi apparatus continually replenish the cell membrane. In summary, the membranous system of the endoplasmic reticulum and Golgi apparatus are highly metabolic and capable of forming new intracellular structures and secretory substances to be extruded from the cell. So, dear listeners, by the way, this educational video is made possible by Prescribon, the makers of e-prescription software for digital prescriptions available freely for doctors on the website www.prescribon.com. Doctors who are interested in this e-prescription can freely avail of its vast collection of more than 100,000 e-prescriptions. The mitochondria extract energy from nutrients. The principal substances from which cells extract energy are foods that react chemically with oxygen, that is carbohydrates, fats and proteins. In the human body, essentially all carbohydrates are converted into glucose by the digestive tract and liver before they reach the other cells of the body. Similarly, proteins are converted into amino acids and fats are converted into fatty acids. Oxygen and the foodstuffs, glucose, fatty acids and amino acids all entering the cell. Inside the cell, they react chemically with oxygen under the influence of enzymes that control the reactions and channel the energy released in the proper direction. Briefly, almost all these oxidative reactions occur inside the mitochondria, and the energy that is released is used to form the high energy compound ATP, that is adenosine triphosphate. Then ATP, not the original food, is used throughout the cell to energize almost all the subsequent intracellular metabolic reactions. Functional characteristics of adenosine triphosphate ATP. ATP is a nucleotide composed of the following: the nitrogenous base adenine, the pentose sugar ribose, and 3 phosphate radicals. The last 2 phosphate radicals are connected with the remainder of the molecule by high energy phosphate bonds. Under the physical and chemical conditions of the body, each of these high energy bonds contains about 12,000 calories of energy per mole of ATP, which is many times greater than the energy stored in the average chemical bond, thus giving rise to the term high energy bond. Furthermore, the high energy phosphate bond is very labile, so that it can be split instantly on demand whenever energy is required to promote other intracellular reactions. When ATP releases its energy, a phosphoric acid radical is split away and adenosine diphosphate ADP is formed. This released energy is used to energize many of the cell's other functions, such as synthesis of substances and muscular contraction. To reconstitute the cellular ATP as it is used up, energy derived from the cellular nutrients causes ADP and phosphoric acid to recombine to form a new ATP, and the entire process is repeated over and over. For these reasons, ATP has been called the energy currency of the cell because it can be spent and reformed continually, having a turnover time of only a few minutes. Chemical processes in the formation of ATP. Role of the mitochondria. On entry into the cells, glucose is converted by enzymes in the cytoplasm into pyruvic acid, a process called glycolysis. A small amount of ADP is changed into ATP by the energy released during this conversion, but this amount accounts for less than 5% of the overall energy metabolism of the cell. About 95% of the cell's ATP formation occurs in the mitochondria. The pyruvic acid, derived from carbohydrates, fatty acids from lipids, and amino acids from proteins, is eventually converted into the compound acetyl coenzyme A, CoA, in the matrix of mitochondria. This substance, in turn, is further dissolved for the purpose of extracting its energy by another series of enzymes in the mitochondrial matrix, undergoing dissolution in a sequence of chemical reactions called the citric acid cycle or Krebs cycle. In this citric acid cycle, acetyl coenzyme A is split into its component parts, hydrogen atoms and carbon dioxide. The carbon dioxide diffuses out of the mitochondria and eventually out of the cell. Finally, it is excreted from the body through the lungs. The hydrogen atoms, conversely, are highly reactive. They combine with oxygen that is also diffused into the mitochondria. This combination releases a tremendous amount of energy which is used by mitochondria to convert large amounts of ADP to ATP. The processes of these reactions are complex, requiring the participation of many protein enzymes that are integral parts of mitochondrial membranous shells that protrude into the mitochondrial matrix. The initial event is the removal of an electron from the hydrogen atom, thus converting it to a hydrogen ion. The terminal event is the combination of hydrogen ions with oxygen to form water and the release of large amounts of energy to globular proteins that protrude like knobs from the membranes of the mitochondrial shells. These proteins are called ATP synthetase. Finally, the enzyme ATP synthetase uses the energy from the hydrogen ions to convert ADP to ATP. The newly formed ATP is transported out of the mitochondria into all parts of the cell cytoplasm and nucleoplasm, where it energizes multiple cell functions. The overall process for formation of ATP is called the chemio-osmotic mechanism of ATP formation. Uses of ATP for cellular function. Energy from ATP is used to promote three major categories of cellular functions. Transport of substances through multiple cell membranes. Example, to supply energy for the transport of sodium through the cell membrane. Synthesis of chemical compounds throughout the cell. Example, to promote protein synthesis by the ribosomes. And 3. Mechanical work. Example, to supply the energy needed during muscle contraction. In addition to the membrane transport of sodium, energy from ATP is required for the membrane transport of potassium, calcium, magnesium, phosphate, chloride, urate, and hydrogen ions, and many other ions, as well as various organic substances. Membrane transport is so important to cell function that some cells, the renal tubular cells for example, use as much as 80% of the ATP that they form for this purpose alone. In addition to synthesizing proteins, cells make phospholipids, cholesterol, purines, pyrimidines, and many other substances. Synthesis of almost any chemical compound requires energy. For example, a single protein molecule might be composed of as many as several thousand amino acids attached to one another by peptide linkages. The formation of each of these linkages requires energy derived from the breakdown of four high energy bonds. Thus, many thousand ATP molecules must release their energy as each protein molecule is formed. Indeed, some cells use as much as 75% of all the ATP formed in the cell simply to synthesize new chemical compounds, especially protein molecules. This is particularly true during the growth phase of cells. Another use of ATP is to supply energy for special cells to perform mechanical work. Each contraction of a muscle fiber requires the expenditure of large quantities of ATP energy. Other cells perform mechanical work in other ways, especially by ciliary and amoeboid motion. The sources of energy for all these types of mechanical work is ATP. In summary, ATP is readily available to release its energy rapidly wherever it is needed in the cell. To replace ATP used by the cell, much slower chemical reactions break down carbohydrates, fats, and proteins and use the energy derived from these processes to form new ATP. More than 95% of this ATP is formed in the mitochondria, which is why the mitochondria are called the powerhouses of the cell. Locomotion of cells. The most obvious type of movement in the body is that which occurs in skeletal, cardiac, and smooth muscle cells, which constitute almost 50% of the entire body mass. Two other types of movement, amoeboid locomotion and ciliary movement, occur in other cells. This figure 1.9 shows amoeboid movement of the cell. The direction of movement is to the right of the picture. Amoeboid movement. See figure 1.9, amoeboid movement of a cell shown earlier. Amoeboid movement is a crawling-like movement of an entire cell in relation to its surroundings, such as movement of white blood cells through tissues. This type of movement gets its name from the fact that amoebae move in this manner, and amoebae have provided an excellent tool for studying the phenomenon. Typically, amoeboid locomotion begins with the protrusion of a pseudopodium from one end of the cell. The pseudopodium projects away from the cell body and partially secures itself in a new tissue area. Then the remainder of the cell is pulled towards the pseudopodium. The figure demonstrates this process, showing an elongated cell, the right hand end of which is a protruding pseudopodium. The membrane of this end of the cell is continually moving forward, and the membrane at the left end of the cell is continually following along as the cell moves. Mechanism of amoeboid location. This 1.9 figure shows the general principle of amoeboid motion. Basically, this results from the continual formation of new cell membranes at the leading edge of the pseudopodium and continual absorption of the membrane in the mid and rear portions of the cell. Two other effects are also essential for forward movement of the cell. The first is attachment of the pseudopodium to surrounding tissues so that it becomes fixed in its leading position while the remainder of the cell body is being pulled forward toward the point of attachment. This attachment is caused by receptor proteins that line the insides of exocytotic vesicles. When the vesicles become part of the pseudopodial membrane, they open so that their insides evert to the outside, and the receptors now protrude to the outside and attach to ligands in the surrounding tissues. At the opposite end of the cell, the receptors pull away from their ligands and form new endocytotic vesicles. Then, inside the cell, These vesicles stream towards the pseudopodial end of the cell, where they are used to form new membrane for the pseudopodium. The second essential effect for locomotion is to provide the energy required to pull the cell body in the direction of the pseudopodium. A moderate to large amount of the protein actin is in the cytoplasm of cells. Much of the actin is in the form of a single molecule that do not provide any motive power. However, these molecules polymerize to form a filamentous network, and the network contracts when it binds with an actin binding protein such as myosin. The entire process is energized by the high energy phosphate compound called ATP. This is what occurs in the pseudopodium of a moving cell, where such a network of actin filaments forms anew inside the enlarging pseudopodium. Contraction also occurs in the ectoplasm of the cell body, where a pre-existing actin network is already present beneath the cell membrane. Types of cells that exhibit amoeboid locomotion. The most common cells to exhibit amoeboid locomotion in the human body are the white blood cells when they move out of the blood into the tissues to form tissue macrophages. Other types of cells can also move by amoeboid locomotion under certain circumstances. For example, fibroblasts move into a damaged area to help repair the damage. Even the germinal cells of the skin, although ordinarily completely sessile cells, move toward a cut area to repair the opening. Cell locomotion is also especially important in the development of the embryo and fetus after fertilization of an ovum. For example, embryonic cells often must migrate long distances from their sites of origin to new areas during the development of special structures. Some types of cancer cells, such as sarcomas, which arise from connective tissue cells, are especially proficient at amoeboid movement. This partially accounts for their relatively rapid spreading from one part of the body to another, known as metastasis. Control of amoeboid locomotion, that is chemotaxis. An important initiator of amoeboid locomotion is the process called chemotaxis, which results from the appearance of certain chemical substances in the tissues. Any chemical substance that causes chemotaxis to occur is called a chemotactic substance. Most cells that exhibit amoeboid locomotion move toward the source of a chemotactic substance, that is from an area of lower concentration toward an area of higher concentration. This is called positive chemotaxis. Some cells move away from the source, which is called negative chemotaxis. How does chemotaxis control the direction of amoeboid locomotion? Although the answer is not certain, it is known that the side of the cell most exposed to the chemotactic substance develops membrane changes that cause pseudopodial protrusion. This figure 110 shows on the left side the structure of cilia. Right hand top picture shows the cut section of cilia. Right side bottom two pictures show forward stroke and backward stroke of the cilia. Cilia and ciliary movements. See figure 110 referred to earlier. There are two types of cilia, motile and non-motile or primary cilia. Motile cilia can undergo a whip-like movement on the surfaces of cells. This movement occurs mainly in two places in the human body, on the surfaces of the respiratory airways and on the inside surfaces of the uterine tubes, that is fallopian tubes, of the reproductive tract. In the nasal cavity and lower respiratory airways, the whip-like motion of motile cilia causes a layer of mucus to move at a rate of about 1 cm per minute toward the pharynx, in this way continually clearing these passageways of mucus and particles that have become trapped in the mucus. In the uterine tubes, cilia cause slow movement of fluid from the ostium of the uterine tube toward the uterus cavity. This movement of fluid transports the ovum from the ovary to the uterus. As shown in figure, a cilium has the appearance of a sharp, pointed, straight or curved hair that projects 2 to 4 micrometers from the surface of the cell. Often, many motile cilia project from a single cell. For example, as many as 200 cilia on the surface of each epithelial cell inside the respiratory passageways. The cilium is covered by an outcropping of the cell membrane, and it is supported by 11 microtubules, 9 double tubules located around the periphery of the cilium, and 2 single tubules down the center, as demonstrated in the cross section shown in the figure. Each cilium is an outward growth of a structure that lies immediately beneath the cell membrane called the basal body of the cilium. The flagellum of a sperm is similar to a motile cilium. In fact, it has much the same type of structure and same type of contractile mechanism. The flagellum, however, is much longer and moves in quasi-sinusoidal waves instead of whip-like movements. In the inset of figure, movement of the motile cilium is shown as a forward and backward movement. The cilium moves forward with a sudden rapid whip-like stroke of 10 to 20 times per second, bending sharply where it projects from the surface of the cell. Then it moves backward slowly to its initial position. The rapid forward thrusting, whip-like movement pushes the fluid lying adjacent to the cell in the direction that the cilium moves. The slow dragging movement in the backward direction has almost no effect on fluid movement. As a result, the fluid is continually propelled in the direction of the fast forward stroke. Because most motile ciliated cells have large numbers of cilia on their surfaces, and because all the cilia are oriented in the same direction, this is an effective means for moving fluids from one part of the surface to another. Mechanism of ciliary movement. Although not all aspects of ciliary movement are known, some details are known. It has been determined that the release of energy from ATP causes the heads of these arms to crawl rapidly along the surface of the adjacent double tubule. If the front tubules crawl outward while the back tubules remain stationary, bending occurs. The way in which cilia contraction is controlled is not well understood. The cilia of some genetically abnormal cells do not have the 2 central single tubules, and these cilia fail to beat. Therefore, it is presumed that some signal, perhaps an electrochemical signal, is transmitted along these 2 central tubules to activate the dynein arms. Non-motile primary cilia serve as cell sensory antennae. Primary cilia are non-motile and generally occur only as a single cilium on each cell. They function as cellular sensory antennae which coordinate cellular signaling pathways involved in chemical and mechanical sensation, signal transduction, and cell growth. In the kidneys, for example, primary cilia are found in most epithelial cells of the tubules, projecting into the tubule lumen and acting as a flow sensor. In response to fluid flow over the tubular epithelial cells, the primary cilia bend and cause flow-induced changes in intracellular calcium signaling. These signals in turn initiate multiple effects on the cells. Defects in signaling by primary cilia in renal tubular epithelial cells are thought to contribute to various disorders, including the development of large fluid-filled cysts, a condition called polycystic kidney disease. Dear viewers, this is the end of part 6 in the 6-part series on the cell and its functions in Medicine for Common Man series. Thank you for watching the video.